I welcome you to the first module, which is to understand the basis of fermentation process when we go back to the history of development of fermentation. The definition: the process of conversion of raw materials into economically important products by use of microbes or microbial enzymes is called as fermentation. These are some commonly used fermented products like curd, bread, etc. The development of fermentation can be categorized under the following heads: the era of discovery of microbes, the process of pasteurization and fermentation, and the discovery of antibiotics. As it is well understood from the above explanation that the conversion of a substrate into a desired product is affected by the activity of microorganism. It is often referred as bioconversion. In some cases, the product is the microbial cell itself, and in such cases, it is called as biomass production. Microorganisms which are often used in fermentation are viruses, bacteria, actinomycetes, molds and algae. These organisms are derived from nature. To use any such organism for industrial production, the strain must be developed. The natural isolate have a low productivity. Fermentations are instantly dependent upon the research and experiment so that the yield can be improved and the problems encountered can be minimized. In the present day life, there are many fermentation products which have found an important place in our daily life, like antibiotics, various enzymes, food products, organic acids and so on. Microorganisms used in industries. The microorganisms can be used in one of the following categories. In the first method, the fermentation is dependent upon a single organism and therefore pure cultures are required. The pure cultures can be stored because storage helps to minimize changes, and it can be stored in glass samples by the process of lyophilization, because lyophilization helps to avoid the number of transfers to a minimum. In the second technique, microorganisms are used in naturally occurring mixtures and under the conditions which are very raw and natural. It is a mixture of organisms that produce the required change in the product. In past, Antony von Leeuwenhoek 1632 to 1723 was the first scientist who saw microorganisms. His observations were recorded in his letters, which were more than 200 to the Royal Society of London till his death. After observation of Leeuwenhoek, scientists did not think much about the origin of microorganism. During the 17th century, the theory of spontaneous genesis or abiogenesis was very popular, which supported that life originated from organic matter. Leeuwenhoek had a view that organisms came from seeds of animalcules. Francisco Redi performed a very basic experiment which explained that spontaneous generation theory was not correct to explain the origin of life. He kept meat protected by placing the vessel covered with a fine cloth to prevent the flies from laying eggs on it, and Spallanzani in 1965 was first to provide evidence that microorganisms do not arise spontaneously in organic matter. There was a series of experiments which did not support the theory of abiogenesis. Finally, the spontaneous generation theory was denied by the experiment of John Tyndall in 1877, who used a box which contained the beam of light to prove that it was empty and the air inside was free from dust and contained no microorganisms. A series of test tubes with the medium was kept inside the box. The broth inside the test tubes remained clear because the box was coated from inside with glycerine to which the dust particles adhered. The germ theory of fermentation. The biological theory of fermentation was first proposed by Cagniard de la Tour, Schwann and Kutzing between 1835 and 1838. Louis Pasteur in 1857 played a very important role in proposing the microbial theory of fermentation. His observation was the production of lactic acid from sugar by different kinds of bacteria. He also noticed the presence of grey color deposits in the fermentation vessels. When these deposits or globules were transferred to another medium containing sugar, yeast extract and chalk, the globules increased greatly in size and number and lactic acid was formed. Pasteur was successful in proving that microorganisms are required for alcoholic, lactic acid, butyric acid and other fermentations. The germ theory of fermentation was laid down by Louis Pasteur, from where the era of fermentation technology began. In the middle of last century, the process of fermentation was thought to be a chemical process. Liebig explained fermentation as a process which was dependent upon a chemical factor which can be changed and decomposed. It therefore causes a chemical change in the basic substance, the degradation of which was then believed to cause a disturbance in the molecule, which had an effect on the final changes associated with the fermentation. The primary reason for the basis of fermentation was thought to be the chemical instability. In 1837, Schwann described the alcoholic fermentation as a process dependent on yeast, but the possibility of presence of a living organism was not accepted at that time. Pasteur worked upon the Schwann's idea on presence of microorganism in alcoholic fermentation. This was the founder experiment which confirmed the observation that a living organism yeast is required for the chemical change that converts sugar to alcohol and carbonic acid. In 1857, Pasteur discovered that there are different kinds of organisms associated with process of conversion of sugars to lactic acid. He explained further that some or the other kind of microorganisms are required for each fermentation. He isolated microorganisms associated with fermentation, then reintroduced them in the nutrient solutions or the appropriate media and observed growth. The natural organisms were removed and reproduced experimentally. The temperature used was 32 to 50 degrees and was found to be optimal for his experiments. He studied the cycles of fermentation and repeatedly removed the organisms from the fermentation cultures and transferred them to fresh sterile media. He again and again repeatedly got the results of fermentation with proper organism present in the appropriate conditions maintained. At the end of all the series of experiments, Pasteur identified the materials that had been the reason for production of the product. He then described the chemical changes which require organisms that were involved in the transformation of sugar to lactic acid. He studied different microorganisms and then described their characteristics. The lactic acid producing organisms were found to be similar and associated to alcoholic fermentation according to Pasteur. Now we know that these two organisms are quite different and have no similarity with one another. 2 decades later, Pasteur studied the number of fermentation materials and also became an expertise in the field of fermentation. In 1858, he discovered the organism associated with fermentation were mold, yeast, bacteria. Pasteur's script was written on the alcoholic fermentation in 1860 and is a classic text available on fermentation and its history on the subject. In 1861, he investigated the butyric acid fermentation and made an important discovery that fermentations can also proceed in absence of oxygen by a rod-shaped organism which was found in with butyric acid fermentation. Pasteur has characterized the bacteria and therefore named it as Vibrio. When he examined the drop of fluid culture containing the tributyrate broth Under the microscope, Pasteur observed that the organism in the margin of the drop became inactive and the organisms present in the middle of the drop were active. He concluded that the organisms became inactive due to presence of air at the margin of the drop, and they were active in the middle because there was no air in the middle part of the drop. Pasteur blew a stream of oxygen through the active fermenting culture and found that presence of oxygen inhibited the action of bacteria. Later on, Pasteur published his research on acetic acid fermentation. Also, he exemplified that this fermentation was completed by microorganisms of genus Mycoderma. Depending upon his studies in 1862 to 1864, in 1868 his studies were published on the complete production of vinegar. Pasteur was invited to solve the problem of the wine industry due to souring of wine. In this, Pasteur described that due to some unwanted organism that came into the wine and altered chemical and physical properties, Pasteur also showed that the undesirable organisms could be removed by partially sterilizing at a temperature below boiling point. The juice from which wine was to be made, this temperature was not sufficient to destroy the valuable property of the juice. The juice can then be introduced with the pure cultures. The process is now called as pasteurization, which was later on also applied in milk industry for the purpose of removal of natural organisms present in milk and the pathogens like tuberculosis, brucellosis and typhoid organisms. There is a data published in 1876 detailed on a 400-page paper which not only described the results but also gave the summary on complete fermentation process based upon living organisms. The era of discovery of antibiotics. In the beginning of 20th century, Emirates obtained antimicrobial substance from the bacteria. This substance was called as pyocinase. It was found to have the ability to produce substance which could destroy disease producing organism. This observation marked the beginning of antibiotic discovery. But then there were no measures to identify the substance which was actually responsible for killing the disease causing organisms, and the study was not fruitful. After a long time of 30 years, accidentally Sir Alexander Fleming came to the discovery of antibiotics, which started the era of fermentation and the production of antibiotics. He was a British bacteriologist who observed a plate which contained the culture of bacteria which have become contaminated by molds. He observed that the bacterial growth was inhibited at the vicinity of the mold colony. He carried this experiment in a hospital at London in 1928. These results were published in the Journal of Experimental Pathology 1929. The microorganism was found to be a strain of penicillin. He did not pursue this investigation further. After a decade, two Englishmen worked upon this fantastic discovery, Cheyne and Florey, who continued his investigation, and an antibiotic preparation was developed for human use. Following this pioneering discovery, later on, a series of antibiotics were discovered which are given in Table 1. After 1880, the sequence of products were then produced by the process of industrial fermentation. There has been an enormous development in the industrial fermentation in last 70 years. The development is both qualitative as well as quantitative. The wide variety of products are nowadays produced by fermentation.